trước khi vào bài học hôm nay thì cô giáo nhắc một tí này. Lần này các bạn sẽ bắt đầu học practical và tutor. Thì cái bài thực hành ấy, nhắc trước không thì lại lúc xin mãi lại không xin được. Thứ nhất là đừng đi muộn. Cái bao em đi muộn cô đã gửi rồi. Cái thứ hai là giả sử các bạn có việc không thể gửi được, giả sử như mà các bạn có em đi, ví dụ như hôm nay em phải đi thi bằng lái xe này, hôm nay em đi viện này, hoặc là hôm nay có vấn đề gì đó về sức khỏe thì các bạn phải gửi trước khi cái giờ thực hành nó diễn ra. Hoặc là giả sử các bạn bị đen một tí hoặc ngã xe chẳng hạn thì các bạn cũng phải gửi và email ngay đến lúc mà có cái tình trạng đó diễn ra thì các cô không vừa nhá, nếu nghiêm trọng quá thì vào viện là có em đi được rồi đúng không? Còn nếu mà không nghiêm trọng đến mức ấy nhưng mà mình có thể đến muộn thì phải email ngay lập tức và cho hai người phụ trách. Hôm nay tôi đã email đây rồi đấy, cho tiếng của cô, cho tôi Hiền. Thì các bạn có thể chuyển sang buổi tiếp theo. Vì sao tôi nhắc thế ạ? Bởi vì là bây giờ thực hành tôi nói thật là không nhắc tổ thực hành là phải gần như là gần như có khi là biếu điểm cho các bạn, chứ còn thực ra những cái report mà các bạn viết thì không bao giờ được điểm đến lúc đó đâu. Nếu các bạn xem điểm thực hành của các năm trước, cho nên là cố gắng là đừng miss buổi thực hành nào cả, rất là phí. Bởi vì là có những bạn 9,8 hoặc 9,9 mấy xin cô cho cô chịu, nó bao em của nhà trường cô không thể nào mà tự động nâng cho các bạn một điểm phần này, một điểm phần kia để cho các bạn qua môn được cả. Đó. Rồi nhắc cái phần thực hành thì thôi, và lúc đi thực hành thì không đi muộn này, làm nghiêm túc này để kiếm được điểm tối đa. Năm nay thì điểm giữa kỳ cũng không cao lắm, cũng không biết điểm cuối kỳ thế nào, nên là các bạn cố gắng đi điểm cái phần thực hành nhé. Hôm nay mình sẽ học không gian chơi, mình kết thúc khoảng 11 giờ được không ạ? Được ạ. Thì hôm nay có 2 tiết không? Alright, so okay, now we come to the last class of these subjects. It's about the subcultures and application of the subcultures. And again, culture here is nuôi cấy tế bào, it's not văn hóa tế bào. So, when you have you ever read any medical studies or in the newspapers? And sometimes you can see the words in vitro or the in vivo. So, these two terms sound quite similar, and it don't look English at all because it's incorporated word, and so it's not surprised that they often make you confused. So here I give you the definitions of the in vitro and the in vivo. So in vitro is mean a medical experiments or the studies that's performed only in laboratories or a test strip. Now, test strip, ví dụ như là IVF is familiar with you. In vitro fertilizations, thụ tinh trong ống nghiệm đúng không ạ? In a test strip, that's what we do an experiment in a test strip. We do the in vitros. And the in vivos is describes the medical experiments or the test that's performed on the living organisms. So, when we do cell cultures, it means we perform an experiment in vivo that we culture the cells in the laboratories, if not in the complete living organisms. So the cell cultures in the process with cells, either prokaryotic cells or eukaryotic cells, are grown under a controlled condition or the artificial environment. So the histories of with the subcultures, it started in the 19th century when the first person is Sidney Ringers. He just develops and discovers, creates solutions, and his name is Ringer Solutions. And these solutions is suitable for maintaining the beings of an isolated animal heart outside the bodies. So this is the purpose of his is create an isotonics. Remember isotonics, hypertonics, and hypotonic solutions. Isotonic solutions, and it's related to the body fluid. Isotonic solution equal to body's fluids, cells are the animal heart can live outside of bodies. And this Ringer solution is typically they contain the sodium chloride, potassium chloride, calcium chloride, sodium bicarbonate, and balanced with the pH. And also glucose, antibiotics, and antifungis. And the next, in 1885, another scientist, he can remove the medullary plate of the embryo chicken. Medullary plate bladder of the ear. He can culture the medullary plates of the embryo chickens and turn it into one saline solution for 30 days. So, it's the first time scientists can establish a principle that tissue could live outside the bodies. It's not really a long time, but 30 days is quite okay. And this is the first time human can raise the tissue outside the body. And next, Harrington's, he's the first scientist successfully grows artificial tissue cultures later on after the work. And by the next, it was really like the boomings of the cell culture and tissue culture started by the Poliers and the Nobel Prize. So Polier, you know this disease? Tại đây đúng không? Yes, so actually the scientist word is poliomyelitis. Poliomyelitis. SARS is one disease that is damage the lungs and kill you by suffocations. But it's not like the coronavirus. It's not attack your lungs only. It attacks also the brains and the nervous system of the muscles. And the muscles do not work. So if your muscles do not work, so you cannot breathe. So when you suffer with the emptiness, have you ever seen the lung irons? And this is the last man on the earth, still ill with the iron lungs now. We cannot breathe by ourselves, and they put the person with the polio disease inside his iron lungs, and these iron lungs can breathe for them. So, this person is, I remember, I don't know if until now he's still alive or not, but 2 years ago he's still alive, and he's more than 70 years old. And the last man on earth lived with the polio disease and with the iron lungs. And before this Nobel Prize in 1930s, they started to culture the virus, the polio virus in the monkey, and then isolate the virus, make the treatment of the polio, but it's not quite efficiency because there is a lot of side effects. Because when you isolate something from animals that's not humans, and so many allergy reactions can happen. And then in 1954, the Nobel Prize is awarded for animals, whalers, and dolphins because they can the polio vaccine, the first virus, they can develop mammalian cells culture techniques. And nowadays, wines are widely used in laboratories, settings for vaccine research, cancer research, protein therapeutics because of this Nobel Prize. They are invent the ways you can culture the poliovirus in the monkey's cells. So we go first, there's tools first about the animal cell culture. The animal cell culture, there's two categories of cells. The first is primary cells, and the second, the established cells. Primary cells, think about that we isolate the cell from our bodies and then we get culture. Established cells is the cell lines in the markets, varies from the companies and use culturing. Our difference, the primary cells, yes, it's obtained directly from the donor tissue. So they have the characteristics of the cell inside our bodies, like these non-transformed, non-immortalized cell direct from the tissue. Immortalized means actually the cancer cells, so we call them primary cells. And these only able to divide finite number of times due to the telomere loss. And after repeats again, 40 to 60 times, depends on the cell lines. The cell will be triggered to the cell mates or sometimes the cell senescence. Cell senescence are no longer divided. So for example, when I work in the laboratories, when I isolate the bone marrows from the mouse, I only culture it for just only 1 week. I kill the mouse, I isolate the bone marrows, cells, culture this all week, do the treatment, have the results, and then you need to trust the weight, you cannot maintain that primary cells in the laboratory. Cells is more efficient when you're the second category of cells is established cells. So it's why whereas the primary cells are obtained directly from the donor tissues, the cell lines can be derived from the clinical tumors, or transforming primary cells. So here's established cells is they able to escape from the normal constraints of the cell cycles, and they can grow indefinitely and extremely used for long-term research. So in my laboratory, we use a lot of established cells instead of primary cells. We can obtain the established cells from 2 ways. First, clinical tumor. We are, actually it's not we, but the humans or the companies, they can isolate the cell lines from the tumors on patients, and then they sell these kinds of established cell line to our laboratory. And the second way, clinical tumors mean we culture the cancer cells. And the second, we can transform the primary cells into established cells by we insert the oncogenes. So when primary cell insert with the primary oncogene will become cancer cells, so we can culture them so many times at once. And the second way, we can add the chemical treatment. This treatment is target. There's target is actually the checkpoint of the cell cycles. And they can make or they can put this kind of cell divide so many times this one and avoid the cell senescence. So viral oncogenes, examples here, A6RE7. So then, this kind of established cell lines can grow so many times at once. We store it in the liquid nitrogen and we use up to 10 or so many years we want. Just maintain the environment, suitable environment for them. One example is one of the first. This is the first cell line that humans isolated. The HeLa cells. HeLa cells are named for the tumor cells obtained from. Actually, the HeLa is the abbreviation of the name of the person who donated the tumors for the science. And these are the patients who died of the cervical cancer. So this person dies by cervical cancer. Actually, the cervical cancer cell lines. And this is the first cell line isolated in the world, and up to now they still used in so many laboratories to perform the research about the cancers, about mechanism underlying for the medical research. And even today, in our laboratory, we have a HeLa cell also in the first cell line in the world used up to now. And for the next, the cell growth environment. So when we want to culture any type of cell, we need to provide the environment that's more close to the environment inside our bodies, right? We need to provide nutrition, oxygen, pH, temperatures, suitable temperatures for them to grow. So for the cell growth environments, first we have two kinds of cells. Adherent culture. We culture it in the this So adherent cell cultures mean the cells stick within the surface. They stick with the surface and they have a monolayers in that surface. Second time we have a suspensive cultures. We cultures in the flask like this. The cell is free floating. It means the cell free floating. In the medium, there's two types of cell. So for example, suspensive cell is roughly the blood cell. So blood cell is free floating in our bloodstream, right? So when we culture the blood cells, we culture it by that way in the flask. They free floating in the mediums. And second, adherent culture is for most of the other cell types, the other cell cultures. And the next, we need to provide the nutrition for them. We provide the appropriate culture media. This is not văn hóa truyền thông, đúng không? This is the môi trường nuôi cấy ạ. scientific word is culture media, môi trường quy tế. So to keep the cell life, it's important to keep the conditions close to the physiology conditions as possible, and therefore we need to supplement amino acids, inorganic soil added into culture mediums. And we take this, one thing we take for the serums of the bovines, so we call them the FBS, fetal bovine serums, is isolated form, yes, for cows. This FBS will provide the vital supplements, macromolecules, lipids, growth factors, and because the scientists do not know the serum exact composition, so it's always subject to a lot of variation between back-to-back experiments. And then second, culture media, they provide amino acids, inorganic soil, vitamin, it is defined mediums. And next, we need to provide the buffer system, and it's often sodium bicarbonate or the HEPAX. It will maintain the pH of the mediums is from 7.2 to 7.4 in 5% of the carbon dioxide. And how we can control the pH? Here we use the phenol red as a pH indicator, where the medium in red, so red is okay, orange or yellow, it means 2 acid. So when it's 2 acids, it will change into yellow. So when you see the medium, it changes into yellows or oranges, means you need to change the medium. It's to a lot of ways, or somehow it has been contaminated with bacteria. But it's too pink, it's too, or the pH may be more than 8, and it's because you check the levels of carbon dioxide in your incubator. And next, you need to keep the correct temperatures. Like when we culture the human cells, we need to keep the temperature of 37 degrees. Mouse, 37, pig, 39, when I counted the pig cell, 39 degrees. Cold-blooded animals is more, is lower temperature for a cold-blooded animal cells. So it depends on the week time of cell you culture. You need to maintain the appropriate temperatures. And very important thing that you need to maintain the environment is sterilized. Avoid bacteria and avoid the virus to attack your cells. So this is the cell we contaminated with the bacteria. You can see so many small dots here. Fungi, okay, this can contaminate with fungi. Or mycoplasma. So you can see the cell with debris here. How to avoid these contaminants? Each unit to work under a laminar flow. You can see in the news, somehow you can see the scientists working under the laminar flows like this. It is to avoid all contaminations for the environment. So the fresh air, the sterilized air will flow continuously to avoid any bacteria and contaminated factors from the environment can go inside this box and contaminate your sample. And next, we can use the antibiotics to prevent the contamination. Antibiotics here are use daptomycin and penicillin. It can prevent some kinds of strains of the bacteria. And the others is not contaminated with the bacteria and from the environment, but there's sometimes by your technical problems, that could be the cross-contamination. So in the yellow sugar here, the first cell lines, and this area is the second cell lines. And sometimes they mix together and it's leading to the first scientific conclusion. So can you answer about this? So it is human cell cultures, and second, bacterial cultures. So bacterial cultures is here is culture the bacteria in the agar plate, and the second is the suspensions, medium also. So here in the agar plate, there's two ways to pour the plate methods. We can put these bacteria in the bottom will be placed, pour the sterile mediums, swirl to mix and incubate it, and then colonize with growth. And or the second way, you can use this specific object to inoculate onto the surface of the agar plate, spread over the agar surface, incubate, and the colonize grows on the surface of the medium. So remember the previous lectures, I give you information that's less than 2% of bacteria and culture. So up to now, there's only more than 2,000 strains. Bacteria can culture in the laboratories, so they are still the mixtures in the humans. And most of them, a lot of cultivars, bacteriums, are the bacteria inside our intestine. So, when we can culture the animals, when we can culture the bacteria, and then we can culture the virus. Sorry. When we can culture the bacteria, when we can culture the animal cells, then we can culture the virus. Because you know that the virus is the parasite, they only can live in the living cells or living organisms. Unlike bacteria or animal cells, can grow on artificial nutrition medium. The virus can be only living in the living blood cells. And when we culture them inside the living blood cell, it's very typical sign that we need to isolate the virus from this kind of cell. Virions is mean the completed virus. So the virions in the liquid medium is get separate on the hot cells, either by centrifugation or filtrations. So here, the fingers can physically remove the anything present in the solution that is larger than the virions. Here in the picture A, you can see the membrane finger. They have a spore side here, and this side is allows only the virus to go through. And here you can see the bacteria in the root state. It's very popular state in the virus the bacteria. And the second is, you can use the finger like this. The light solution can filtrate through the finger like this. You can see the red particles in the animal cells, the blue in the bacterial cells, and the green here in the virus. So you can see how we can isolate the virus from this solution. So with the filter pore size of 5 micrometers, it can allow bacteria and virus also can go through. But with the pore size of 0.2 micrometer, it allows only virus can get through, and you can obtain the solution that contains only the virus on it. So when we can culture the virus, we can identify and diagnose some of pathogen virus in the clinical specimens, produce the vaccines, and we can perform some basic research study only. So here on the left, you can see some flux is used in cultures of the human and animal cells for virus culturing. And on the right is here the animal cell for culture. And here if we use the bacteria. For culture the virus. And here you can see this is the level of the virus is just increased. When you see left 4-9 of bacteria in here, you equalize. When it's more clear, it means that this more virus kill the bacteria. This small dot here is called line of bacteria. And when the number of virus increase, the number of colonies of bacteria is decreased. And is this in vitro cultures? What about the in vivo culture? In vivo culture virus, here we can use the X. So the X is ants for breakfast only, so they can the source of the common flu shot. And here most of the influenza vaccine, the vaccine are manufactured or annual flu vaccine program in culturing the hen's eggs. So the cell within the chicken eggs used to culture different types of virus, and the virus can be replicated in various locations within the eggs. And you know that we can use the egg to culture the coronavirus or not? Have you ever heard that we use eggs to produce the vaccine for coronavirus? Yes or no? The axis will be the answers. People need to find the alternative ways of the eggs to produce the coronavirus vaccines, because the receptor The act is not suitable for the coronavirus to stick it on it and to invade and manipulate it. Actually, the receptor is ACE2, as I remember. So for the produce of the coronavirus, is we need to grow the kidney cells from monkey kidney cell, to produce the coronavirus vaccine. So this is the applications of the animal cell culture. We can test the effects of drugs on cell growth, test the therapeutic effects of drugs on the cancer cell life. We can use for tissue engineering, produce the vaccine and monoclonal antibodies. We can study the replication and cell lines of the virus. We can culture the embryo culture results, perform the embryo culture results. Production of therapeutic proteins, or you can use as large water to avoid the use of the animal for preliminary studies. So the first we perform the in vitro cultures, and if it works, it trends to in vivo culture. So in my laboratories, we worked first, second, and third. These three terms we do in the laboratory. We test the cancer drugs, test for the treatment of inflammatories, we test the treatments of the hyperglycemics, we test for the treatment, to test the treatment of osteoporosis. And if you have interest in animal cell cultures, you can come in the second floor, room 0.5. It is laboratories for animal cell cultures there. Xin một phút để quảng cáo nha. Và các bạn ở bên TMAS NST, ở trường mình nếu bạn nào mà có hứng thú với việc nuôi cấy tế bào động vật và thử các thuốc hoặc là các hợp chất mà có tác dụng chữa bệnh ở trên tế bào động vật có thể đến phòng tầng 2, phòng 205 ạ. Ở đó đang nuôi cấy rất là nhiều các dòng tế bào khác nhau, và nhóm của cô thì đang tìm kiếm các thuốc có thể chống lại bệnh ung thư, chống tăng sinh đường huyết để chữa bệnh tiểu đường ạ, có thể chống bệnh loãng xương ạ, hoặc là chống kháng viêm. Đó, và đáng tiếc thì có thể đến phòng này ạ. Nhận sinh viên từ năm 2 ạ. So next, about the plant tissue culture. Năm 2 lên cứ lựa xem hoa để làm cho năm 3. Cells are stimulated to the animal cell cultures. When we want to culture the plants, we need to provide the appropriate environments for the plants. Cells we need to provide nutritious medium, of course, to avoid the contaminations and the septic conditions, and also the aeration of the tissue. Here, we can take these small piece of the apical meristem. Cái này là đỉnh sinh trưởng đúng không ạ? Apical meristem in the plant, and we put in agar place. Then we put the hormones auxin, cytokinin, and then a single cells can form from a whole plant. It's just because the plant cell have two characteristics. First, totipotencies, and the second, plasticity. So the totipotency is the ability of the cell that can generate the whole plant from the single cell of tissues by continuously division. Meristem, apical meristem là điểm sinh trưởng, còn meristem là mô phân sinh. And then the second characteristic of the plants is plasticity. So when the plants tissue cultures, they exhibit very high degree of plasticity. It means that one type of tissue or organs can be initiated from the other type under the influence of the chemicals. And by this way, the whole plants can be generated from the single cells or tissue. So, what is applications of the plant tissue culture? It is micropropagations. So, it's in very high rates, production of disease-free plants, cost effective. And here is a process that you can generate for, it's not like the previous pictures. Under the laboratory is much more complicated. Here is staminate, and then you can transform by culturing the medium. This can form the columns. The colors is the undifferentiated mass of tissue that can grow into complete plants. So then it generates into embryo. And next you can grow these kinds of embryos, root formations, and when you have a complete plant, you can make its growth. This is how you can produce a complete plant under the laboratories. Actually, these pictures I take from my thesis or my bachelor thesis. I do the micropropagations in my bachelor lab. And this is how I generate a complete cacao tree from a stem nut. The next application of the plant tissue cultures. We can take advantage, or we can call it the secondary metabolite productions. Or you can take the compounds produced under controlled conditions, or quality products with the consistent production of normal compound production is less of the times, and it's make the secondary metabolites less expensive. So here is the application of the hairy root. You culture the plant cells of tissues induced of the hairy root, and the hairy roots can transform root culture into a type of plant tissue that's used to study metabolic process, produce valuable secondary metabolites, recombinant proteins. And here they have a high flow rates and generate stabilities, produce high level secondary metabolites, produce stable levels secondary metabolites over long times, scaling up in plants, bioreactors, or metabolic engineering, or enhance the second by metabolic engineering. You can, or humans can enhance the secondary metabolite production. Is the plant genetics engineering. So examples of these, you can use the hair to produce our innings. or so many other people that useful to use a drug. Này kỷ niệm bạn đã bị sốt xuất huyết rồi. So we can use the hair to produce large numbers of the secondary metabolites. And for plant genetics ingredients, there's one issue that is GMOs, genetically modified organisms. How we can perform a GMOs? There is one bacteria strain, we call it Agrobacterium tumefaciens. They're very special because they contain one kind of plasmids. Remember plasmids in the previous lecture? TI plasmid. We cut the TI plasmids, we cut out one of the genes, we insert into the TI plasmids, and we have recombinant TI plasmid dipole. And then we insert back into the bacteriums. This kind of bacteriums and the specialty of the TI plasmids is they can insert the DNA carrying the poison gene, insert into the chromosome of the plant. And after they transform the TI plasmid, after they transform the genes, the wanted genes, into the plant, then we culture the plant cells, and then next, by the step in the previous slide as I introduced to you, this can grow or regenerate new plant cells with the new trait, wanted trait. So do you know what is the pros and cons, or what is the advantage or disadvantage of the GMOs? Are you agree with GMOs? You use the product made of GMO plants or not? Any issues of the GMOs? Any comment about the GMO you would care? Whatever the product you use, you don't care about this label that is non-GMO or GMO products. Do you think it's good for the humans or not? example for neighbors? That's no. Why yes and why no?
Thế ra là do bọn nó đạo đức. Ra là biến đổi gen thì trên con người là bị cấm hay sao? Những người mà liên quan đến chuyên môn thì người đã bị bắt.
Thực ra là không phải là trên con người cấm, mà là nó phải được qua rất là nhiều bước, nên là cái đấy chắc chắn là an toàn và được thực hiện.
Tại tôi thấy mấy cái vụ nuôi cấy với cả chọn lọc gen bị bắt hết.
Tế bào cell chạy của nó thực ra nó không có bây giờ làm được cell rồi làm trên một cái sống là kiểu thì biết về chuột bạch chuột đấy, nói chung là thì biết về chuột sống thì có bài cái đấy thì nó về tranh cao còn cells.
Cái này đang làm gì đấy?
One question. With the following cannot be used to culture the viruses? So what is the correct answer for this question? B is right.
Because viruses need a host.
Yes, virus needs the host. We cannot culture the virus in artificial mediums or artificial environment, right? So A, C, and D, they are all
Have a host.
contain the living stuff. Tissue contain living cells. Embryo is perfect for the virus, like we can culture the virus in the eggs, chicken eggs, right? And the animals is perfect environment to culture the virus. But if it's only liquid medium, that is without the living cell, we cannot culture the virus. And we go to some questions to revise for the lesson number 8. Okay. Scientific patients, apoptosis, stem cells, cancer cells, and also the prokaryotic cells. 3 lessons. We have no games, but we're going to explain question by questions, and it's time for you to think more time to think and make your correct answer.
Hả? Có nghĩa là điện tử khó. Đừng gọi là khó nhé. Chắc là khó rồi. Không ma cha được rồi. fill in blank. Ở đây chụp trước đáp án. Cô bắt giơ tay ấy. Kiểu giơ tay đấy ha. Điện tử.
Từ từ cô nhắc lại nhé, tất cả các thứ liên quan đến lịch học, sinh nghỉ, danh sách các thứ hỏi thư ký nhé, đừng email cho cô thì sau cô lại pass cho thư ký xử lý thôi. Đến bây giờ các bạn vẫn hỏi lớp này lớp kia chưa có lịch thì hỏi cô, hãy hỏi thư ký khoa.
So we are doing some multiple choice section for the lecture 9 about autosis, cancer cell and stem cells. So each question we will have 1 minute to think, and I will call 20 to answer the question.
Good.
So we can start with the first question now.
Program cell data use for the to be
program đen cái sự sao chứ
tinh and control cell lead to cancer. Bốn cái là ăn control rồi, ba là gì? Cái ba vision display. Một là program, hai
Một là
damage.
Apoptosis is the process of program cell death. Apoptosis is used to rid the body of cells that have been damaged beyond repair. Apoptosis also plays a role in preventing cancer. If apoptosis is for some reason prevented, it can lead to uncontrolled cell division and the subsequent development of cancer.
Then your answer is true.
Correct. Vậy là chú.
Sorry to modify the mentioned question that is may appear in your final exams.
Cái này ở trong final.
Question number 2.
Anh bị giết người. Anh bị giết người.
So question for what are the hallmarks of the necrosis? 1 minute.
Hallmarks. ra đây nhưng mà mạng kém quá. Chỉ là it's a very Hallmark là dấu ấn á. Dấu ấn của Necrosis là gì? Hallmark of her disease. Pattern during khó đây. Để tổ du sinh lắm chết.
Your answer is B or not? Yes.
Hỏi tử à? Rất không nên tra cái này. Cái gì đấy? Trông nó lại ghê thế này. Nó hoại tử cái gì mà trông ghê.
So in the question 6, you will have an image that describes the
Okay.
two types of cell death, and you are required to
Necrosis.
to define which is A, which is B, and which is C.
Đi là gì? Kiểu cái này á. Cái này á. Chắc là cái này. Đúng không? Không, chả biết.
Nhưng mà hỏi group of position là gì?
Kiểu necrosis là cái phân ra đúng không? Necrosis hoại tử ra mà apoptosis là cái B. Cái B là cái gì? B là normal cell. Chắc thế.
Thì hoại tử chắc
Hoại tử là cái bên phạm C rồi.
cái kia chắc
Hoại tử là cái C.
chỉ có necrosis và apoptosis thì chắc cái là lại là apoptosis.
Ghê. Rồi.
Thì đã hỏi tại sao cái gì?
Tôi đã dốt sinh nên là bị hỏi thì cũng chết. Ra cái quá. Thì anh chán lắm, tiếng Anh ố gì lắm. Vừa mới thi hôm trước. Có phải qua không? Không qua lại phải không? Anh phát. Yên tâm hơn tí. Ố gì rồi.
So we have 3 words to answer these questions. First word is apoptosis, second word is necrosis, and the third word is normal cells. So you can attribute A, B, and C, and then my teaching assistant is forgot to insert the words here.
Yeah, đúng rồi. Đấy, như nói vừa không ấy. Cái bên A là apoptosis, cái bên phải bên C là necrosis, giữa là nổ xéo. Cô vừa nói xong là cái anh kia quên không cho 3 cái từ ấy vào để điền. Nói chung là đúng.
phải tự
So,
chuẩn bị áo lab,
so my activity B would be
chuẩn bị áo
from
lab.
cell.
What?
A would be
Cái gì đấy? Gmail mới à?
C would be apoptosis.
Ai khó đấy. Sao mà tự chuẩn bị
So can you describe a little bit about apoptosis,
gì ạ?
please?
So the process C of apoptosis will be the death of cell caused by it's the program death caused by fragmentation.
Okay, so in the apoptosis process, we will find some fragmentation. It's called the
Chứ không phải chuẩn bị áo
apoptosis
lab.
body.
Sinh viên tự chuẩn bị. Cậu đéo ơi, mất đâu. Áo lab. Cốt mua ở đâu? Tí lên hỏi cô. Tí đi hỏi
37.
Tí đi hỏi thư ký của cô. Wow, lab cốt.
Or we're talking about necrosis.
Mẹ gửi email này gửi sớm chứ gửi muộn lắm đấy.
Thing is B. Cell contains agarose by macrophage.
No.
Full with statement này còn thêm S nữa thì is A nữa thì
The answer is E.
là hai cái Necrosis.
Who has other answer?
Necrosis, I think it must be E.
So the question is asked about the fall. Which is fall, not true. So the correct answer here is E, DNA fragmentation. So A surrounding cells are infected, so because the necrosis will cause inflammations. Cell shrinkage, and when the cells occur necrosis, all cells will shrink and burst. C is always occur, and when all the content of the cell is exposed to the environment, our body will send the macrophage to eat this waste.
Chắc hôm nào phải thử tải gamma ly thì thấy hai cũng dùng sợ hôm nào cũng thử gamma ly.
And the next question, which statement of truth when talking about the hypothesis? Star 110 suggests by neighboring cells, here, significant inflammation reaction occurs, plasma membrane integrity is maintained. Cell lysis or cell burst?
Cell kỹ cell nên A sai B C thì không biết. Còn được 8 phút nữa thôi, thử xem có 8 phút nữa được không. 8 phút thì cũng đang chỉ được.
answer is I, but this question has one more answer.
A and B.
So the answer of this question is A and C.
C. That's my main.
For this question, I think that the cell content digester when neighboring cell is not really precise, because it's just ended up by the other cell and they can reuse the content inside this package to reuse them. It's not to digest them. Significant inflammation reaction occurs is for necrosis, not apoptosis. Plasma membrane integrity is the popular hallmarks of the apoptosis. So the plasma membrane integrity and also the nuclear membrane integrity is the characteristics of the apoptosis. Cell lysis, no. Cell burst, no. Is for necrosis.
Cô Duy hay ngon. Tôi nghi ngờ là cô không biết gì hay ngon đấy. Cái gì đấy? À, cái này á.
I think this an easy question, and you can call some student to answer it like that. It's quite easy.
Là A đúng không? Vì
Even
là
you
A.
don't study about the apoptosis, you can guess from these five words.
Nó đấy. Mày là B là đấm nhau. Đúng không chú ăn phun? Đâu rồi nó không muốn trả lời. À, sẽ ưu tiên đứa ra nhìn điện thoại. Sẽ ưu tiên đứa ra nhìn điện thoại. Ưu tiên đưa mic vào. Hộ mà ra nhìn thiết bị điện tử. À đúng. Đấy.
Vì cái thiết bị điện tử người ta không nhìn thấy trên mạng.
Thường là có nhìn máy tính với điện thoại là bị gọi, nhưng mà nhìn thẳng ý khi bị gọi. Đấy thôi thì chắc là
Nhìn thấy đây rõ. Chứ không nhỏ nhỏ.
đấy thôi, một đáp án. Cá lĩnh chi.
Thôi coi được cô này, được cái đây câu này các bạn phải đoán được là nhìn mouse power gì ạ? Bàn con chuột ạ. Under gì nhỉ? Embryonic là phôi ạ. The apoptosis shape the organ during the development. Sao ạ? Nó định hình các cơ quan, xác định hình dáng các cơ quan trong quá trình phát triển. Cái bài này ngay mở đầu apoptosis cô đã nói về role của apoptosis rồi. Trong quá trình phát triển thì cái bàn của các động vật có xương sống là nó có màng quanh các ngón giống như màng bơi đúng không ạ? Và trong quá trình phát triển thì apoptosis sẽ phân tách các ngón tay hoặc là ngón chân trong quá trình phát triển phôi để tạo ra một cái bàn tay hoặc bàn chân hoàn chỉnh. So in this question, the A in the correct answers. Apotheosis take the organ during the development. Next questions?
Oh shit. Cô đang chỉ ôi rồi. Cái này là cái gì á? Cái này là cái gì? Cô hỏi cô này hơi khó.
Căn bản là không hiểu.
Mày có hỏi cái cô đi em đi ông trả tiền này.
Kiểu tính.
Nào, bạn nào không học môn này ít nhất nhớ cho cô hai từ apotheosis và necrosis nhé. Còn mấy cái tuberculosis rồi bệnh lao rồi silicosis thì bỏ qua nó đi một cái. Cái câu này chỉ là 50-50 thôi, chọn
Ồ,
nào.
50-50. Aposis. Chắc thế.
Chỉ.
Oh,
Giải đôi đi cũng.
I think it's a
Nói đi thì mà nói to lên.
nói thôi mà. I think it's a
To lên.
không nói to được nữa. Em nói thế thôi. Nếu anh bắt anh nói to nữa thì chịu. Chết mẹ em. Wow, lạp. Cột. Tôi chả được của gì thì này.
I think it's necrosis because you can see that the edge of the cell has some fluid that is like going out, so it must be necrosis that has more probably.
Không biết.
Um,
Em đi tìm
You mean you see the leakage of the saboteur?
and yes,
là cô chỉ đầy nhiều.
with the right arrow.
Không
No. So the answer here is apotheosis, not necrosis.
Thì đúng như thế thì không thể giải
The
thích.
black arrow here is the nucleus. They are fragmented. So in the apotheosis, we'll see the DNA fragmentation, and we also see that the plasma membrane here is still maintained. So this cell is undergoing the apoptosis.
Yeah.
Here, this picture is necrosis because the cell is burst.
And you can see that so many part of the cell has been disintegrated, right? Oh, last question. The ability of the cell to relocate to a distant organ is called Yeah, this is metalysis process what kinds of cells can be moved to other areas in your bodies. So here is the last questions, and this is the last lesson that I teach in the auditoriums. And I have some words for you. Tôi nói tiếng Việt cho nhanh nhé. Có hai cái mà cô nhắc các bạn là xin đừng bỏ lỡ trong môn SEO của cô. Thứ nhất là bài practical, hai bài practical rất quan trọng với các bạn về mặt điểm số. Cái thứ hai là bài tutorial trước sau, bài cô sẽ revise các bạn từ bài đầu cho đến bài cuối và cho các bạn làm một số câu hỏi tổng hợp như là một số câu trắc nghiệm và cái cách mà cô làm sao để cô ví dụ trong bạn có trắc nghiệm và là số cô đổi đáp án như thế nào các bạn làm được. Nói chung là hai cái đấy thì đừng bỏ lỡ. Cái nữa là hôm nay cũng là buổi cuối cùng rồi thì cô có một vài lời muốn nói với lớp. Thứ nhất là cảm ơn các bạn đã tham dự, ít nhất là có thêm một nửa số sinh viên đã tham dự ngày hôm nay. Năm nay có một cái hơi cái nói là thực ra là hơi đặc biệt của cô, bởi vì là năm nay thực ra là cô đi dạy khi mà cô vừa mới sinh bé được có 2 tháng thôi, cho nên là nếu như các bạn nhớ là bài đầu tiên ấy, tự nhiên đến cái cô plasma membrane tự nhiên đâu cytoplasm, cô nhớ mãi mới ra được cái từ là chất nguyên sinh. Thực ra là rất là đầu óc nó hơi mây một chút và cũng không thời gian ở lại để giải đáp câu hỏi cho các bạn. Ngoài ra thì có một số cũng cho các bạn link Google để các bạn gửi câu hỏi. Lắm lúc thực sự là cô đã soạn thư để trả lời cho các bạn rồi đấy. Mấy hôm sau thầy nó đang ở cái chế độ Grab và vẫn chưa trả lời cho các bạn được. Thì có một số cái nó gọi là nó không được như mọi năm. Thì mong các bạn cũng là thông cảm và chúc các bạn là ôn thi thật tốt này, đạt được điểm cao nhất ở trong bài practical, ôn thi tốt và không hy vọng gặp lại các bạn ở trong test đâu.
Em bị hỏi đoạn lab code chưa hiểu.